Hi everyone. In this short video presentation, I'll explain you how innate immunity works. I'm going to explain you step by step in the following order. Number one, barriers, which include physical, chemical, and microbiological barriers as a first layer of defense in innate immunity. Number two, antimicrobial proteins and peptides as a second layer. Number three, cellular sentinels as a third layer. And number four, acute phase proteins as a last layer of defense in innate immunity. I'm Dr. Mo. I'm Associate Professor from Newcastle University.
Okay, so let's start with the first layer of defense in innate immunity, which is the barriers. These barriers include physical, chemical, and microbiological barriers. In terms of physical barrier, we consider skin as our first line defense because the skin, which is the keratinized outer layer, forms an airtight waterproof seal that pathogens cannot easily penetrate. Mucous membrane of respiratory, gastrointestinal, and urogenital tract traps microbes in the sticky mucus. Cilia in the airway acts as a mucociliary escalator, beating upward to push trapped pathogens out of the lungs. Furthermore, tears, saliva, and urine physically wash microbes out of the body cavities. Among chemical barriers, body pH plays an important role in certain organs. Acidic fluid in the stomach and vagina kills most of the swallowed and introduced microbes. Lysozyme, found in tears, saliva, and sweat, actively cleaves peptidoglycan cell walls of bacteria. Antimicrobial peptides, in short AMPs, molecules like defensins and catalycidins puncture microbial cell membranes. Sebum, secreted by skin glands, contains fatty acids such as oleic acid, which can lower skin pH and inhibit bacterial growth. Lung surfactants such as lipoproteins can bind to pathogens and assist in their clearance. Microbiological barriers of innate immunity consist of trillions of harmless microorganisms which are normal flora or microbiome of the body. They naturally inhabit the skin and mucosal surfaces where they serve as a biological first line of defense against pathogens. These commensal microorganisms prevent pathogen colonization through competitive exclusion by occupying epithelial binding sites and through nutrient competition by consuming available resources required for microbial growth. In addition, they produce antimicrobial substances such as bacteriocins and short-chain fatty acids, which inhibit pathogen survival by lowering the local pH and constantly damaging invading microorganisms. Okay, let's move on to second layer of defense in innate immunity. There are two components in this layer: antimicrobial proteins and peptides. There are several antimicrobial proteins that protect you against pathogens. This protein kills microbes through enzymatic destruction or cutting off their nutrient supply. The essential ones you need to know are complement proteins, lysozyme, lactoferrin, transferrin, and psoriasin. The most important antimicrobial proteins that you must know is called complement proteins. These proteins work across your entire extracellular environment in your body, including your gut, lungs, and urogenital tract. They patrol the bloodstream constantly to detect pathogens, or they may leak from the bloodstream into tissues to fight localized infections. There are more than 30 complement proteins, and the majority of them are produced by the liver. They have three main functions. One, killing of bacteria by opsonization and phagocytosis working together with phagocytic cells. 2. Induce inflammation and dilatation of blood vessels, thereby drawing neutrophils and other immune cells to the site of infection. And 3. Lysis of bacteria by formation of a ring-shaped hole, which is called membrane attack complex, or MAC in short. I'll explain you in detail about these proteins in the next video. Other antimicrobial proteins that you should know are lysozymes. These are the enzymes highly concentrated in tears, saliva, mucus, and the granules of the phagocytic cells. It attacks the structural integrity of bacteria, breaking the chemical bonds in the peptidoglycan cell wall that causes bacteria to burst from osmotic pressure. Another antimicrobial proteins are lactoferrin and transferrin. Lactoferrin can be found in the mucosal secretions of saliva, tears, and neutrophils, and transferrin in the blood. Both proteins execute nutritional immunity by tightly binding and sequestering free iron. Because bacteria need iron to replicate, these proteins effectively starve and stop the growth of microbes. The last antimicrobial protein that you should know is psoriasin. It is secreted by keratinocytes of the skin, particularly on the face and hands. It selectively sequesters zinc, a critical nutrient for bacterial survival. This protein specifically targets and kills Escherichia coli and other gram-negative bacteria. There are 3 major antimicrobial peptides that you should know. First, defensins. Defensins include alpha and beta defensins, and they are produced by neutrophils, macrophages, and epithelial cells of the skin, respiratory tract, and GI tract. They possess a net positive charge that binds to negatively charged microbial membranes. They insert themselves into the bilayer, forming pores that cause cell lysis and death of microbes. Catalycidins are another antimicrobial peptides that you should know. They are stored in neutrophils secreted by mucosal epithelial cells in response to infection. They attack bacteria, envelop viruses, and fungi by disrupting their cell membrane. They also neutralize toxic bacterial endotoxins. Dermcidin. They are secreted continuously by eccrine sweat glands onto the surface of the skin. It forms zinc-dependent ion channels in the microbial membranes, disrupt the cell's electrochemical gradient, and kill the pathogens. Okay, let's move on to cellular sentinels, the third layer of defense in innate immunity. This layer consists of specialized leukocytes that patrol tissues, engulf invaders, and coordinate inflammatory response. The leukocytes include phagocytes, granulocytes, cytotoxic cells, and antigen-presenting cells. In terms of phagocytes, we have neutrophils and macrophages. Neutrophils destroy pathogens through phagocytosis and by releasing enzymes and reactive oxygen species, or ROS. Moreover, neutrophils can undergo a unique form of programmed self-death by which they burst and release neutrophil extracellular traps, or nets, which are sticky web-like structures that trap invading pathogens, holding them in place to kill them later. Macrophages. They are the major phagocytic cells that engulf large volume of debris and pathogens. They secrete pro-inflammatory cytokines, interleukin-1, interleukin-6, and TNF-alpha to recruit other cells and clean up dead neutrophils to promote tissue healing. Another key function of macrophages is to act the antigen-presenting cells that display degraded pathogen fragments on the surface so that T and B lymphocytes can recognize them and initiate an adaptive immune response. Granulocytes include eosinophils, mast cells, and basophils. Eosinophils, they specialize in fighting parasitic infections such as helminths. They bind to the parasite and release highly toxic proteins such as major basic protein or MBP. MBP binds and disrupts the structural integrity of the cell membranes of the parasites, resulting in pore formation and cell lysis. Mast cells and basophils. Mast cells reside in tissues such as skin and mucosa, and basophils circulate in the blood. When activated by injury or allergens, they rapidly release granules filled with histamine and heparin. These actions dilate the blood vessels and increase permeability, allowing other immune cells to rush into the affected area. Another group, cytotoxic cells, which play a major role in clearing body's own compromised cells rather than attacking external microbes directly, and it is called natural killer cells or NK cells. NK cells patrol the body, rapidly identify and destroy virus-infected cells and early-stage cancer cells. They kill these cells by releasing perforin to punch holes and granzymes to induce apoptosis. Another killing mechanism of NK cells is by expressing surface proteins such as FASL or Fast ligand and TRAIL that bind to the DEF receptors on the target cell and initiate apoptosis. The last group of cells in our innate immunity is antigen-presenting cells, which consists of dendritic cells and macrophages. I've explained you macrophages just now. Let's focus on dendritic cells here. Dendritic cells reside in an immature state within peripheral tissues, particularly boundary environments such as skin, where they are called Langerhans cells, as well as the gut and respiratory mucosa. Although these cells phagocytose and kill pathogens, their main function is antigen presentation. Once they engulf a pathogen, they degrade it, migrate through the lymphatic system to the nearest lymph node, and present the processed foreign antigens to T lymphocytes to launch the adaptive immune response. Okay, let's move on to the last defense layer of innate immunity, which is acute phase proteins. Acute phase proteins, in short APPs, are a class of serum proteins whose plasma concentrations increase or decrease by 25% or more during an inflammatory state. These proteins are primarily synthesized by the liver, although extrahepatic tissues such as adipose tissue, lung, intestinal epithelial cells, and macrophages also produce specific types to assist and immediate local defenses. APPs are divided into positive APPs, which increase to destroy pathogens and regulate tissue repair, and negative APPs, which decrease to conserve raw amino acids for the positive response. Among positive APPs, C-reactive protein or CRP acts as a soluble pattern recognition molecule that binds to phosphocholine expressed on the surface of dying host cells and bacteria. Once bound, it activates C1q complement pathway to clear the target via opsonization and phagocytosis. Of note, CRP is the most widely used marker for systemic inflammation. Another APP is serum amyloid A or SAA. It binds to bacteria to help phagocytes identify and destroy them via phagocytosis and recruits immune cells to the site of infection or tissue damage. That is called chemoattractant. The third APP that you should know is fibrinogen. It is cleaved by thrombin to fibrin, forming a sticky structural mesh or clot. This physical barrier traps invading microbes, stops bleeding, and provides a scaffold for tissue healing. Among negative APPs, albumin is the one of the important proteins that you should know. During inflammation, the liver downregulates albumin synthesis to save amino acids for making positive APPs. This reduction alters blood osmotic pressure, allowing fluid and immune cells to exit blood vessels and enter inflamed tissues more easily. Another negative APP is transferrin. The liver also downregulates transferrin synthesis and secretion, thereby reducing total ion transport capacity in the bloodstream and helping sequester vital ion resources away from circulating pathogens. So the net effect of negative APP is to promote inflammation and immune cell recruitment via albumin and starving microbes of ion via low transferrin. So in summary, I have explained you three barriers which include physical, chemical and microbiological barriers as the first layer of defense in innate immunity. Four antimicrobial proteins which include complement proteins, lysozyme, lactoferrin, transferrin and psoriasin, and three antimicrobial peptides which include defensins, catalycidins and damcidins as a second layer of defense. In the third layer of defense, four types of cellular sentinels which include phagocytes, granulocytes, cytotoxic cells and antigen presenting cells were explained. In the last layer of defense, I explain you the acute phase proteins which are composed of positive and negative phase proteins.
I hope this video presentation is helpful for you. If you like it, please subscribe and share. Thank you and have a nice day.